What if you could brew a cup of tea that also delivered your annual flu shot?
Hmm. Interesting thought.
Or maybe cultivate the absolute rarest medicinal plant compounds right there in a lab, leaving, nature completely untouched.
Okay. Yeah.
Let's really get into this because today we're exploring the pretty fascinating world of plant tissue culture.
It really is remarkable. And it's a field that, well, it sounds incredibly high tech, doesn't it? But it actually has quite a deep history behind it.
Right.
The materials you've pulled together give a fantastic overview, really tracing it from those initial, almost foundational experiments
Uh-huh.
right up to the sophisticated medical applications we're starting to see now.
Exactly. And you've given us this really comprehensive set of sources. So our mission here is to pull out the most compelling, the most useful insights for everyone listening.
Basis.
We're going to sort of journey through the key historical moments, get a handle on the essential techniques, figure out what these lab-grown plants actually need to thrive.
Right. The nitty-gritty.
Yeah. And then look at some truly mind-bending applications like creating new medicines and even those edible vaccines you just mentioned.
And looking at the bigger picture, plant tissue culture really gives us this incredibly precise way to guide how plants grow and develop. It bridges that fundamental plant science with real tangible benefits for health, for agriculture. It's about taking a very direct hand in how plants work.
So where did this all kick off? I mean, it wasn't like these super sterile high-tech labs just appeared one day, was it?
Oh, definitely not. Not at all. The early days were really marked by painstaking investigation, lots of trial and error.
I bet.
Our sources really highlight researchers like Gothard and White back in the 1930s and 40s.
Okay.
Gothard, for instance, he managed to culture cambium cells. That's like the growth layer in trees.
Right, under the bark.
Exactly, from things like willow, poplar, maple, even elm and black locust. He actually got these cells dividing and growing for quite a while. That was a huge step.
Just imagine trying to keep those delicate cells going outside the tree. That must have been incredibly difficult. And White, he was working with tobacco, but a specific kind.
That's right, yeah. It was actually tumor tissue from a tobacco hybrid. And he also achieved continuously growing cultures around the same time.
Okay.
But what's really key about both their work is they started figuring out the nutrient soup, basically.
Ah, the recipe.
Exactly. What basic food, what conditions these plant tissues needed just to survive and, multiply in a lab flask. They were the real pioneers working out those fundamentals.
So they were like the first chefs in the plant tissue culture kitchen, figuring out the basic ingredients. What came next? What were the big breakthroughs after that?
Well, the field kept pushing forward. A lot of focus stayed on refining those nutrient media. People like Raghavan, Torrey, later Norstog, they did important work on media for culturing really young embryos.
Embryos, like plant seeds starting out?
Sort of, yeah. Culturing them very early on. It was vital for understanding those initial stages of plant development. But then, in the 50s, came the really pivotal work by Skoog and Miller.
Skoog and Miller. Okay, that sounds important. What did they do?
They brought in the idea of hormonal control. Big step. They found that by carefully tweaking the balance of different plant hormones.
Plant hormones, right? Like auxins, cytokinins.
Precisely those. By adjusting the ratios, they could basically tell the tissue what to do.
Tell it what to do.
Yeah. I mean, a certain mix might encourage roots to form. Another mix could trigger shoot development.
Wow.
Or another balance might just keep it as a mass of undifferentiated cells. It's called callus.
Callus, like a blank slate of plant cells.
Exactly. So this ability to steer development in vitro in the glass was a massive leap. One of the sources has a great figure showing how auxins help cells get longer, gibberellins push stem growth, cytokinins drive cell division.
That's amazing. So fine-tuning chemical signals lets you direct the plant cells. And then Morishige, his name pops up everywhere in this field. What was his contribution?
Morishige was huge for making tissue culture practical, really practical. He saw its potential for quickly multiplying plants, especially in horticulture, making lots of copies of a good plant variety.
Scaling it up.
Exactly. He developed standardized methods and a very famous medium called MS medium.
MS medium, right. Morishige and Skoog.
That's the one. It worked well for a huge range of plants, ferns, flowers, even fruit trees. He basically took it from a pure research technique to a viable way to propagate plants on a larger scale.
Okay.
Imagine making thousands of identical copies of a prized orchid or something just from a tiny piece. He helped make that a reality.
So moving from basic discovery to like mass production almost, huge step. And what
Right.
about regenerating a whole plant from just one mature cell? That sounds like science fiction.
It really does, doesn't it? But Braun actually did that. In 1959, he proved the concept of totipotency in plants.
To Tippins.
Yeah, the idea that a single adult plant cell still holds all the genetic information and the potential to grow into a complete new organism.
Incredible.
And then in the 60s, Cocking figured out how to isolate protoplasts, that's plant cells, with the cell wall stripped off.
No wall. Why do that?
Well, he used enzymes from a fungus to digest the walls, getting large amounts of these naked cells. Think of it like taking the shell off an egg carefully. It opened up totally new ways to do genetic manipulation fusion experiments.
Yeah.
Things you couldn't do easily with the wall intact.
Wow, okay. So from culturing tissues to directing them with hormones to even working with individual cells without their walls, the field was really unlocking things quickly. And you mentioned work starting in India, too.
Yes, absolutely. The source notes that research kicked off there in the mid-1950s, largely thanks to Panchanan Maheshwari at the University of Delhi.
Ah.
He's often called the father of plant embryology in India, actually. So yeah, this push was definitely a global scientific effort.
Okay, great. So that gives us a really good sense of the history. Now let's shift gears. If someone's listening and picturing maybe bubbling beakers or something really complex and intimidating, what does a plant tissue culture lab actually look like?
That's a good question.
Mm-hmm.
It's maybe less bubbling beakers and more controlled cleanliness.
Okay.
Ideally, a lab is organized into separate areas. You'd have a general workspace, maybe a larger room for preparing the nutrient media, washing glassware, sterilizing things in a hot air oven.
A hot air oven for glassware?
Yeah, it gets it really hot and dry, kills microbes, and purifying water, usually with a distillation unit. Then there's the culture room itself.
Right.
That's where the plants are actually growing in their containers.
Yeah.
It needs really precise control over light, temperature, humidity, very stable conditions.
Makes sense.
And then there's a smaller, absolutely critical room or area for aseptic work. That's where you do the transfers, putting the plant tissue into the fresh medium, usually inside a special cabinet.
The laminar airflow chamber you mentioned earlier.
Exactly that. It's probably the most crucial piece of equipment for avoiding contamination.
So how does it work? What makes it so special?
Well, it creates a sterile working zone. It pulls in air from the room, runs it through a pre-filter for dust, and then forces it through something called a HEPA filter.
HEPA. High-efficiency particulate air.
We got it. This filter is incredibly fine. It traps nearly all bacteria, fungal spores, tiny particles from the air.
Wow.
Then this ultra-clean air flows smoothly in parallel streams, that's the laminar part, across the work surface. It constantly pushes room air away, preventing contaminants from drifting into your sterile cultures while you work.
Like an invisible shield of clean air.
Pretty much, yeah. And the procedures are strict. You turn it on well beforehand, wipe everything down with 70% ethanol, maybe use UV light first. All about keeping things sterile.
Makes sense. So besides that and the hot air oven, what other key bits of kit are in there?
Oh, quite a few essential tools. You need a refrigerator, obviously, for storing chemicals that are sensitive to heat, like some vitamins or hormones. The distillation unit, or water still we mentioned, that provides the pure, sterile water that's the base for all the media. It basically works by boiling water, collecting the steam, which leaves impurities behind, and then condensing that pure steam back into water.
Right, like nature's own purification cycle. What else?
You absolutely need accurate weighing balances. Some chemicals are added in tiny amounts, so precision is key. A pH meter is crucial too, for getting the acidity of the growth medium just right.
pH matters that much?
Oh, definitely. Plant cells are sensitive to it. And then there's the autoclave.
The pressure cooker thing.
Yeah, kind of a high-tech, high-pressure steam sterilizer. It uses moist heat under pressure, usually about 121 Celsius, 15 psi to kill absolutely everything, even tough bacterial spores. Vital for sterilizing the media, tools, containers.
Got it. Safety must be a big deal with that.
Absolutely. You have to be careful, make sure containers aren't sealed tight, never autoclave flammable solvents or strong acids. There are strict protocols.
Okay, so that covers sterilization and basic prep. What else is needed for actually growing and looking at the cultures?
Well, you need basic working tables for media prep, maybe a simple heater or hot plate to melt the agar. That's the stuff that makes the medium gel-like.
Agar, right. Like in Petri dishes.
Exactly. Microscopes are obviously essential. Different kinds, simple magnifying glasses, standard light microscopes for looking at cells, and stereoscopic microscopes too.
Stereoscopic. Like 3D.
Yeah, they give you the 3D view, which is great for manipulating tissues or looking at larger structures without complex slide preparation.
Yeah.
Very handy.
Okay.
For liquid cultures, you'd use an orbital shaker to keep things gently mixed and aerated.
Right, stop cells settling out.
Precisely. And centrifuges are often used. Different types, low speed, high speed, sometimes refrigerated ones, basically for separating things based on density, like spinning down cells from a liquid medium.
Like a high-speed salad spinner for cells.
That's a good way to think about it. And then depending on the research, you might see more advanced gear like PCR machines.
For DNA work.
Yeah, amplifying specific bits of DNA. And gel electrophoresis units to separate DNA fragments by size. That comes in more for genetic analysis or confirming transformations.
So a real mix of standard lab gear and some very specialized stuff for handling living plant tissues at the micro level. I guess with all this precision, keeping records must be incredibly important.
Oh, absolutely critical. The sources really hammer this home. Meticulous record keeping in a proper lab notebook or field book is non-negotiable.
What kind of details?
Everything. Experiment name, date, all the materials used, the scientific principle behind it, the exact step-by-step procedure, any calculations, everything. There are even rules, like numbering pages, never tearing pages out, how to correct mistakes properly.
A scientist's detailed diary, basically. And safety, I imagine that's a constant focus with all the chemicals and sterile procedures.
Huge focus. Lab protocols always spell out safety measures, how to handle chemicals correctly, like always adding acid to water slowly, never the other way around.
Right, I remember that from chemistry class.
Yeah. Proper labeling of everything, safe disposal of waste, wearing personal protective equipment, lab coats, gloves, sometimes eye protection. And aseptic technique is paramount. Constant vigilance to avoid contamination. Wiping down the laminar flow hood with ethanol, handling sterile items carefully. It's ingrained.
Makes sense. You have to be meticulous. Okay, so we've covered the history, we've peeked inside the lab. Let's get to the heart of it. How do you actually do plant tissue culture? How do you grow these tissues?
It all starts with the explant.
Explant.
Yeah, that's just a small piece of tissue you take from the parent plant to start the culture. It could be a bit of leaf, stem, root, flower bud. Depends on the plant and what you're trying to achieve.
Okay.
And the very first absolutely critical step is surface sterilization. You have to kill off any bacteria or fungi living on the surface of that explant.
Otherwise, they grow faster than the plant tissue.
Exactly. They'll just overwhelm the culture.
Yeah.
So it usually involves a series of washes. Tap water, maybe some detergent, an antifungal solution, then sterilizing agents like ethanol for a short time, or maybe dilute bleach or mercuric chloride.
Mercuric chloride? Sounds nasty.
It is potent, yeah. Used carefully and rinsed off thoroughly afterwards with sterile water. The goal is to kill surface microbes without killing the plant tissue itself. It's a delicate balance. The manuals have specific recipes for different plants, like pineapple or banana.
So a super thorough cleaning process for the plant bit. What then? Where does it go?
Once it's clean, the explant is carefully placed onto the nutrient medium in a sterile container, like a jar or a petri dish. And that medium is its entire world.
The nutrient soup we talked about.
Exactly. It provides everything the plant tissue needs. Inorganic salts providing macro and micronutrients, like the calcium chloride mentioned in the popular MS medium recipe.
Right, murashige and skoog.
Yep. Then a carbon source, usually sucrose, for energy. A gelling agent, typically agar if it's a solid or semi-solid medium. And those crucial phytohormones we discussed, auxins, cytokinins, maybe gibberellins.
The growth regulators.
Precisely, to control the growth and development. Plus vitamins, things like glycine, thiamine, and maybe myo-inositol. It's all mixed in precise amounts, the pH is adjusted carefully, and then the whole medium is sterilized, usually in the autoclave.
It really is a carefully crafted diet at the cellular level. And you mentioned different kinds of cultures you can grow from these explants.
Yes, several main types. Callus culture is very common. That's the unorganized growing mass of undifferentiated cells.
The blank slate.
Right. You can induce callus from many explants using the right hormone balance. It can be grown on solid medium or sometimes in liquid as a suspension culture. And callus itself can sometimes be embryogenic, meaning it can form embryos and potentially whole plants, or non-embryogenic. The non-embryogenic kind is often good for producing secondary metabolites, those useful chemicals.
Ah, okay. What else besides callus?
Suspension cultures, as I mentioned, single cells or small clumps floating and dividing in liquid medium, usually kept moving on a shaker. Then there's organ culture, where you're trying to grow or maintain an organized structure.
Like tiny organs.
Sort of. Things like meristem cultures, using the very tip of a shoot to grow a single virus-free shoot, or root cultures, shoot cultures where you multiply existing shoots, embryo cultures to rescue or study embryos, even anther and pollen cultures.
Anther and pollen. Why those?
To create haploid plants, plants with only one set of chromosomes, very useful for breeding programs. And finally, protoplast cultures, using those cells without walls we talked about earlier.
So a whole range, from messy callus to organized bits and pieces, all growing in the lab. How do you go from one tiny explant to, say, hundreds or thousands of plants for farming or nurseries? That's micropropagation.
Exactly. Micropropagation is the process for rapidly multiplying plants in vitro. It generally follows distinct stages. Stage I is initiation.
Getting started.
Right. Establishing that clean, healthy culture from your explant. Stage 2 is multiplication. That's where you encourage lots of new shoots or propagules to form, often by dividing clumps and moving them to fresh medium that promotes shoot growth. You could really bulk up numbers here.
Okay, making copies.
Then stage 3 is rooting. You take those individual shoots and put them on a different medium, usually with more auxins, to encourage roots to develop.
Makes sense. Need roots.
And the final step, stage 4, is acclimatization, or hardening off. This is crucial. Those lab-grown plantlets are quite delicate, used to high humidity and sterile conditions.
Right. They've had an easy life.
Exactly. So you have to gradually get them used to the real-world, lower humidity, normal soil microbes. It's often done in phases, maybe first in a mist chamber or a greenhouse with special potting mix, then slowly reducing the protection before they can survive in the field.
A gradual transition to the outdoors.
Precisely. The source materials give really good detailed examples for pineapple and banana. The specific media used at each stage, the hormone combinations, how long each stage roughly takes. It's a well-defined process for many commercial crops now.
It sounds like a really systematic, almost industrial process for plant production, but all starting in the lab. What about things going wrong? Contamination seems like it would be a constant headache.
Oh, it is. It's the bane of tissue culture. Contamination by bacteria, fungi, things like Aspergillus penicillium, yeasts, sometimes even algae. They can get in and just grow like crazy on that rich nutrient medium, killing the plant culture. The sources show pictures, actually, macroscopic views, microscopic views. It's not pretty. Prevention is absolutely everything.
Yeah.
Strict sterile technique, keeping the lab environment clean,
Right.
regular cleaning of incubators and shelves, careful handling in the laminar flow hood. It's a constant battle.
Constant vigilance against microscopic invaders. Okay, so shifting focus slightly, one of the really exciting applications is using this for medicines, right? Pharmacognosy.
Yes, that's a major area. Plant tissue culture, especially callus and suspension cultures, can be a great way to produce secondary metabolites.
Secondary metabolites, those are the useful compounds.
Often, yes. They're chemicals the plant makes that aren't essential for its day-to-day survival, but might be involved in defense, attraction. And many have valuable medicinal properties. Think about alkaloids, flavonoids, terpenoids. Lots of drugs originally came from plants.
Right.
So by growing the plant cells in culture, sometimes we can get them to produce these compounds, maybe even more efficiently than the whole plant in the field. You can sometimes tweak the culture conditions, the nutrients, hormones, even add elicitors to stress the cells slightly to boost production. Figure 4 in the source nicely shows callus being used this way.
So you could potentially grow the drug-making factory part of the plant in a bioreactor instead of harvesting whole plants from the wild.
That's exactly the potential. It's huge for conservation, especially for rare or endangered medicinal plants, and it offers a more controlled, potentially sustainable supply chain.
That's fantastic. How do you get the compounds out of the cultured cells then?
Various extraction techniques are used, similar to extracting from whole plants. Maceration is as simple just soaking the dried ground cells in a solvent. But it can be slow and use lots of solvent.
Okay.
Soxhlet extraction is more efficient. It uses a clever setup to continuously wash the material with fresh, hot solvent, faster, uses less solvent, but the heat can sometimes degrade sensitive compounds.
Right.
And then there's supercritical fluid extraction, often using carbon dioxide under high pressure at specific temperatures where it acts like a liquid in a gas. It can be very selective and clean, leaving no solvent residue. The best method depends on the specific compound you're after.
Fascinating range of options. From nurturing cells in a dish to pulling out complex molecules, it really does feel like we're just scratching the surface. And that brings us to maybe the most futuristic sounding application. Edible vaccines. Plants as tiny factories making vaccines.
Yeah, it's a really cutting-edge area. The basic concept is genetic engineering. You engineer the plant so that it produces an antigen.
Antigen being the molecule that triggers an immune response.
Exactly. A specific protein or part of a protein from a virus or a bacterium, for example. If you can get the plant to make this antigen in its edible parts, say in a banana or a tomato or lettuce leaves, then theoretically just eating that plant material could deliver the antigen to your immune system, acting like a vaccine.
Wow. No needles.
Potentially, yes. The big appeal is cost effectiveness and ease of delivery, especially in developing countries or remote areas where cold storage and trained personnel for injections can be major hurdles.
Makes sense. And tissue culture helps make these plants.
Absolutely. Tissue culture techniques are essential for creating the genetically modified plants in the first place, and then for rapidly multiplying them, scaling up production so you actually have enough vaccine plants to make a difference. It's a key enabling technology for the whole concept.
Just amazing. Eating a banana to get vaccinated. It sounds like pure sci-fi, but it's actually being worked on.
It is. There are still challenges, of course, dosage control, stability, public acceptance, but the potential is definitely there. It really shows the power of plant biotechnology.
Okay, let's try and wrap this deep dive up with some key takeaways. We've covered a lot of ground.
We certainly have.
We traced that really cool historical journey, didn't we? From Gawthrette and White just trying to keep cells alive, through Skoog and Miller figuring out hormonal control,
Right, a huge step.
to Morishige making it practical. We got a glimpse inside the lab, that super clean, controlled environment.
The importance of sterility and equipment like the autoclave and laminar flow hood.
Yeah, and we explored all the different ways you can grow plant tissues, callus, suspension, organ cultures, the whole micropropagation process.
From initiation right through to acclimatization.
And then the application, especially in medicine, using cultures to make those valuable secondary metabolites.
A sustainable alternative, potentially.
And ending with the really mind-blowing idea of edible vaccines.
Plants as bioreactors for health.
So for people listening, what this really shows is just how much we can now guide and manipulate plant life, right, for all sorts of benefits.
Absolutely. From boosting food production to developing new healthcare solutions. It's about understanding and harnessing the power within plant cells.
Yeah.
In a really controlled way. It's quite remarkable, actually.
It really makes you think about what could come next. So maybe a final thought to leave everyone with, considering this incredible precision we now have, manipulating plants right down to the cellular level, what other maybe currently unthinkable feats of biological engineering might actually become possible down the road?
That's a big question.
It is. It feels like this deep dive has opened a door onto a world just packed with potential, and the discoveries definitely aren't stopping anytime soon.